Well, I now introduce Dan to present our third presentation, the next generation tools, pipelines, cluster, and PEPMatch. Thank you, Dan.
Thank you, Nina. Hey, everybody. So I'm Dan. I work in the Peters Lab as a bioinformatics specialist. So I'm going to be demonstrating an example of using the pipeline feature for the next gen tools that Jay touched on. So some of this beginning stuff will be a little bit redundant to what Jay talked about. So yeah, we're going to be talking about next gen site. And so I'll be using this example of these dengue virus envelope proteins. So we have 4 of them. These are the full sequences for the proteins. And for the MHC1 predictions, we'll be selecting between 8 and 10 peptide lengths. And we'll be using the 27 allele panel and these various prediction methods that Jay touched on. So you can input your sequences into the box here, along with your peptide length, and then selecting the 27 alleles using the allele finder button. And again, selecting these prediction methods. So again, everything that we've already touched on, and click run, and there you have your peptide table. And I want to also touch on that in these tabs, you have the peptide table, you have the net MHC-PAN allele distance, which is basically the net MHC-PAN algorithm has a way to see how far away alleles are from each other in terms of how the method will use for predictions. So these are standard alleles, so using further or non-standard alleles will have a further distance from each other. And the sequence table is the original sequences that you input. And so yeah, back to this table, the original table from the results. You can do the filtering again. Let's say we filter on median binding percentile and immunogenicity score, and then total processing score again. And so you have these filtered, so you have your rows shrunk down, and then you can save this table state. And once you've done that, now you can actually pipe on these results to another tool. So we're going to go from T-cell prediction class, what we just did, pipe it to the epitope cluster analysis tool, and then pipe those results to PEPMatch tool. And so the reason that this we wanted to demonstrate this is because it's a very common thing that people want to do, including us when we use our own tools and also external users. They typically want to use results from one tool and pass them to another tool. And so the next-gen tool site makes this much more easier for users. So on the left-hand side of the panel, you're going to have this plus box, and you're going to click your tool you want to pipe to. In this case, we're going to pipe to the cluster tool, and we're going to select this is the peptide table that we showed earlier and the peptide column. So that would be this column here, which is all the peptides that were produced by the MHC binding prediction tool. And then you're going to select your parameters. So it's all streamlined too. It's not going to even take you to another page. It'll just open up at the bottom, and you'll put in your parameters for the cluster tool or any of the other tools that you select. So in this case, we want to limit our clusters by 70% sequence identity. So this would be clustering peptides that are at least 70% similar. And here you could select like a length parameter. In this case, we're not using any. And then there's various cluster methods of ways to cluster the peptides together. We recommend this one as a cluster break for clear representative sequence, so you can actually show the representative sequence for that cluster. And I'll show that in a second. And click run. And this is another useful feature as part of the pipeline, is that it'll give some warnings sometimes. So let's say you have a bunch of peptides in here that are duplicates. So it's just warning you that it's going to remove these duplicates for the piping to the next tool. So this is the output for the cluster analysis. And so yeah, the peptide table. And again, you can filter on these columns. So here we have the alignment of the cluster of the peptides within the cluster, and the consensus sequence, which is the consensus amongst all these peptides at each residue position. And again, you can filter and save and download this. And there's also a visualization tab up here at the top, where you can see each one of these dots is a peptide and the clusters that the peptides belong to. And you can also click this drop-down menu to select various clusters that have been produced by the tool. And so let's say we want to filter on just using the consensus sequence. So you can click on consensus here at this drop-down, this filter button. And now you have, again, this these yellow bars in the table that show that we've done some filtering. And so now we can save this table state again. So now we just have these consensus sequences. And now we can pipe on those peptides to another tool. And in this case, we're going to be selecting PEPMatch, which is a tool that does a search within a selected proteome to find the peptide at a certain mismatch residue or certain mismatch threshold. So you can select PEPMatch, and again, we see that the peptides were selected. And then here's the parameters again for PEPMatch. So you're going to select the number of mismatches you want to allow. So these are residue substitutions, and then the proteome. We have various proteomes for the next-gen site, like mouse and dog and pig. You can also select the best match. So best match based on the number of mismatches or some other parameters. And then also, or you can just get all the matches. And then there's also a checkbox here to include peptides that did not get a match anywhere in the proteome. And so click run again. And so here's the output for the PEPMatch tool. You can see there's various, again, there's various columns that you could filter on, like the protein ID from UniProt, the protein name, the gene it comes from, the number of mismatches for the input sequence and the match sequence, and the residue positions that those mismatches are at. Yeah, you can save and download those as well, and then plus potentially pipe them to another tool. So here's a documentation for the cluster tool and also for the PEPMatch tool. We have documentation on all the tools that are available, and also on how to actually implement the pipelines like I just went through again. And I'm going back to these slides that Jay showed earlier to show that we have these CEDAR-specific tools that you can also use. The PEPX tool right now you can use for the pipeline feature. And again, we have all these tools that are planned for release and other plans. And so you can imagine that as these tools get added to next-gen site, we'll have like a network of tools that there'll be a massive amount of combinations of these pipelines you can generate. And then the other big bonus to it is that you can save those pipelines via the links and then share them amongst your collaborators or colleagues. So it doesn't get too complicated for you to keep track of what you're doing. So yeah, test it out now. It's available on the next-gen site, as I showed. And so we hope that it's very useful and intuitive for you. Thank you.